Sometime around 2008, a mysterious disease started killing off the Netherlands fire salamanders. 3 years later, 96% were dead. The disease turned out to be Batrachochytrium salamandrivorans, or Bsal for short, a relative of the chytrid fungus Batrachochytrium dendrobatidis, or BD, that has been implicated in the decline or extinction of some 200 frog species around the world. Scientists think Bsal originated in Asia and spread to Europe through the pet trade, and they believe it's only a matter of time before it gets to the US, the world's hotspot of salamander diversity, where nearly half of all species may be susceptible. Now, scientists are in a race against time to find the fungus as soon as possible after it gets here, in the hopes that quickly enacted quarantines may stop, or at least slow its spread. My name is Mike DiGirolamo, your host for Mongabay Explorers, a special podcast series about some of the most recent reporting from Mongabay.com's global team. Join me for a deeper discussion of one such project, where I'll explore the issues with experts on the front lines of the looming salamander pandemic to find out what we know now and what is being done to keep North America's and the world's salamanders safe. Today on the podcast, I sit down with former Mongabay reporter and current energy reporter for Business Insider, Benji Jones, and wildlife disease ecologist with the United States Geological Survey, Daniel Grear, to talk specifically about what Benji has described as searching for a needle on a haystack, except the needle is invisible and the hay stretches for thousands of miles. This is a massive undertaking that no small amount of effort, time, and resources would be able to complete, but enough government agencies are pooling their resources to search for and monitor the appearance of this dangerous fungus. Today, I talk with both Jones and Grear about the search for this fungus, the insidiousness and difficulty in finding it, and what future efforts we can expect. I understand that you work for the U.S. Geological Survey, and you have been involved with the testing of salamanders?
Yes. Working with a group of amphibian researchers within USGS, and then a larger interagency collaborative group that also includes university NGOs, set out to identify what's risky for salamanders. And then we've been one of the central labs to go out and do sample collection and receive samples and do diagnostics for this new salamander chytrid fungus.
Can you tell me, just provide with me some context, when did the USGS start getting involved with this effort to test salamanders for this chytrid fungus?
It started shortly after, the science, the amphibian, the conservation community realized what a huge threat the Bsal potential pandemic, now probably is full-on pandemic, was going to be. So that would have been in around 2015, 2016. And so the first step was to try to assess the risk. How bad could it be if it was going to get here? How could it get here? Where would it be the worst? And we used that then to reaching out to collaborators, working with USGS scientists at different science centers across the country. Starting in 2016, went out with a goal of, based on this risk assessment of where we think the biggest impacts would be, the most likely place for this new fungus to get released in North America, sample as many of those places as we could. And so over 2 years, we received over 11,000 samples, primarily different salamander species from across the country. We did all of the diagnostic testing here in our molecular biology lab.
I'm sorry to interrupt, and that's in Madison, Wisconsin, correct?
Yep. So that effort, those 11,000 samples came in over 2 years. We ramped up a lot of our capacity to handle and get those tested in the lab, get the data processed and communicated back out to submitters. And thankfully, we did not detect it in that big sample. But now we're at the point where we're starting to put together this information from, not detecting it in a lot of samples, starting to get together with the various researchers who are doing susceptibility trials, any kind of solutions, any kind of mitigations that we could do for there to think about what we're going to do next.
11,000 samples is quite a lot of tests, and you might expect that after such a concerted effort that if Bsal was present, you would most certainly find it, which is encouraging since it hasn't been found to date, yet that doesn't mean that it couldn't necessarily be present yet remain undetected. Would you say that if it was here or if it was present somewhere in the United States, do you necessarily think that the current testing efforts we have would find it.
In certain circumstances, if it somehow got released relatively recently, but spread quickly to a lot of different areas, we think the current testing probably would have detected it. But if it was a situation where it had been either for some reason not spread very fast, not caused a big impact to where it was initially released, so something where people would not have noticed a lot of dead, sick animals, we don't have a whole lot of confidence that we would have necessarily detected it if there's not a lot of it out there yet.
If the disease wasn't detected after 11,000 samples, and it's technically possible for it to be present without being detected, it would be reasonable to be a bit concerned. But before you get alarmed, Grear articulated quite well how the current testing efforts are focused in on areas of the country that have the greatest concentration of biodiversity of salamanders, and ultimately how these efforts can effectively monitor the appearance of this fungus.
Through our testing work, we continue not to find Bsal, which is encouraging, but the caveat is there's a lot of salamanders out there in North America. The Fish and Wildlife Service did put in some rules to prevent the import of certain species of salamanders, but there's always the continued risk that, somehow the fungus is going to get here. So one of the things we're finding is that we can continue to test, but we're really just working around the margins of trying to be more certain about whether North America is Bsal free or not. We can hit and focus on what we think are the most important places, but there's a lot of places we just don't have the capacity to look.
And can you tell me what some of those places might be?
It's really focused on where salamander diversity is greatest in North America. So it's along the Pacific coast, and then from the southern Appalachians up through the Alleghenies. Those are just absolute hotspots for number of species, abundance of species, very unique species of salamanders in North America.
Can you tell me a little bit about, there was a USGS heat map which showed the high-risk situations of Bsal entering the U.S., and on that map I saw like a concentration of it like around the New York City, New Jersey areas, and other areas where there seemed to be lots of population density. Is that because of those are hotspots for the pet trade, or is there another specific reason?
It's both of those. It's where people are, and it's where our major international ports of entry for the pet trade is. We're making the informed assumption that if Bsal does get to North America, it's going to come in the pet trade. That's all the evidence seems to point that's how it moved into Northern Europe. So that heat map, what that represents is risk as a compounding of where the international imports of pet amphibians come, where we think there are susceptible hosts out there, and where we think the pets might get released, which really is just related to where people are.
And are you currently, I know you said you're focusing your testing efforts on the Pacific coast and also in the Appalachian areas to the Alleghenies. Is there any kind of concerted testing effort in and around those communities, those urban hotspots?
Not to speak of. There is, the Bsal task force has discussed ways to try to approach or do testing, both as a way to detect, but also to potentially help the pet industry with demonstrating that it's safer or not. But we haven't really made any inroads there. And as far as focusing on wild populations where they're close to people, not really since that big first push for surveillance where we aimed for those hotspots. So if you're interested, also, we should have a scientific publication coming out soon with the results of all that.
That publication is slated to be released in the summer. However, Daniel stressed that due to COVID-19, there may be a delay in getting that information to the public. Still, even with these efforts, finding Bsal isn't necessarily going to be easy. To learn more about this, I spoke with Benji Jones.
Something that's concerning always with diseases like this is just like salamanders that could be asymptomatic. Right now, you could have people with COVID who are like silent carriers. The same thing can happen with salamanders. And so the reason that's a problem is that if you're a carrier and you're not showing symptoms and you're not dying as a result, that means that you can be constantly carrying around this, infecting other salamanders.
And even if Bsal is detected, like COVID-19, that could very well mean it has spread to lots of areas already.
I think one problem is that once we find it, it's likely somewhat widespread already, just because a lot of these salamanders are in pretty remote areas, which on the one hand could mean that they're less likely to be infected, but if they are infected, we probably won't come across them very easily, right? So like someone literally needs to cross paths with a salamander if it's not a deliberate testing effort. So that's the one problem, is that if you find a salamander with a Bsal infection with chytridiomycosis, then it likely is already spread pretty far. And then I think the other one is just that Bsal spreads really quickly.
Because Bsal is a waterborne fungal pathogen, it doesn't just infect salamanders, but it can adhere to the feet of birds. Even beavers can be potential carriers, making this already deadly pathogen all the more insidious.
The other issue with Bsal is that it's a fungal pathogen that's in the water, and so it's just really difficult to contain because streams are not bound by like political boundaries, for example, and it's difficult unless you're draining them to kind of eradicate it completely. And so it can spread really easily through waterways. If one salamander that has it goes from one pond to another, you can see how quickly it would spread. And then, as I mentioned before, one type of this pathogen, spores, can adhere to the legs or the feet of birds, and so they could also be spreading it. So I guess bottom line is that it can just spread pretty quickly. And it is enduring, so it can last in the environment for, I believe, like a week or more, at least one type of spore of this pathogen.
And that's somewhat different than past pandemics that we've had, such as BD.
What I learned through my reporting is that BD only has one type of spore that is motile, so it has like basically like a little propeller on the back of it, and so it swims around in the water, the spore does, and it doesn't last as long. So this type of spore Bsal also has, but Bsal has this other spore as well that does last longer. So the main difference is that BD has only one type of spore that is shorter lived, and Bsal has two, one of which can live a little bit longer.
Perhaps the one main advantage that we have over Bsal, unlike the previous amphibian pandemic of BD, is the fact that we are aware of it before it has hit our nation. Both Jones and Grear explain the unique opportunity that this has given us. Switching over to another pandemic, can you tell me, how has the outbreak of BD influenced your research in testing with Bsal, if at all?
Well, it's, for better or worse, it's given us an idea of what could happen. So in some sense, we've seen this happen before when there's been and only really recently the amphibian disease community has really started to fully account for the spread and the impact of the global pandemic of BD, what we think of as the frog chytrid fungus. So yeah, in some sense, it gives us some idea of what could potentially happen. And with that, it seemed to be a very rapid spread, although as a science community, as a research community, as a conservation community, we were chasing that. We didn't know it was coming until after it was well into its, global spread. For Bsal, we at least with this, big testing in North America, all the research and testing across Northern Europe where Bsal impacts were first detected, we are more confident that we're at least somewhat ahead of the pandemic curve with Bsal, at least in North America. So what we can learn certainly from BD is a little bit about the biology because they are closely related pathogens, but there's still a lot to learn about how each of the fungus interacts differently with frogs and salamanders.
Jones felt similarly optimistic.
We've sort of been here before with chytrid fungus and its impact on frogs and other amphibians. And so we kind of knew what the scale of devastation could be. And so on the one hand, that makes it way scarier because the risk that we could wipe out like a large chunk of salamander populations. But on the other hand, scientists that were working on that are already experienced with a disease like this. And so there is the ability to prepare to some extent, which they've done. We've never really seen any kind of preparation for a wildlife disease like this, to my knowledge. I mean, certainly not with BD, certainly not with white nose syndrome.
So, armed with this knowledge, Grear and many USGS scientists have been hard at work testing thousands of salamanders. I asked Grear to explain this process a bit more in depth, what specifically they are looking for when they test, and whether or not there are additional testing methods being researched. And as it turns out, there are actually quite a few different testing avenues that they are looking into.
So the goal of swabbing the salamander, so rubbing a small, usually polyester or rayon Q-tip all over their bodies, is to pick up little bits of the fungus, because the fungus infects the skin, and in order for the fungus to transmit, it needs to release its spores out of the skins of the animal hosts. So swabbing the skin surface is a good way to try to detect it. And then when those samples get sent to us, what we do in the lab is attempt to detect the DNA, the fungal pathogens. And the testing in the lab is very sensitive. We can test small amounts, down to what we would consider probably 10 or so of these really tiny spores of Bsal or BD. And it's specific enough that we can differentiate whether or not in animals we can detect the BD fungus or the Bsal fungus. It's all very similar diagnostic process to, the issues that are going on currently with the human coronavirus testing and pandemic. It's the same types of techniques, same types of, trying to find a bit of the pathogen in a swab where it lives on a person or a salamander, and then taking it back into the lab and trying to detect its DNA.
This is just kind of a curious question I have. Would it be possible to somehow also detect it on another animal, say like on the foot of a bird? Or is that something that you guys consider in your testing efforts, or are you simply keeping it just strictly testing salamanders?
Right now we're focusing on salamanders and frog and toad species too. When there's the opportunity and folks were able to catch them, they swab them as well, because we don't really know what the full ability of the Bsal fungus is to infect different amphibian species. There's a lot of great work at different group of researchers are doing susceptibility trials, so ideally in the next year we'll have a much better idea of what amphibian species can get infected. As far as testing things like, ideas that pop up from time to time are being able to test the feet of birds, or beavers are a species that, move around a lot in aquatic systems. It's just a lot easier to get your hands on the amphibians than it is any of these other species that aren't really infected but could have, a little bit of fungus attached to their toenail or fur. One testing avenue we are working on, and a lot of people are also trying to dig down on, is being able to filter the fungus out of the water, because part of its life cycle does require it to be free living in the water for a little bit.
So almost like potentially being able to just take a sample from a pond or a lake and seeing if the fungus is present in the water itself.
Yes. And so that involves pushing a lot of mucky water through filters. And a lot of what we're working on, what I think a lot of other amphibian disease researchers are working on, is how do you make sense of that when you're suddenly moving from the skin of something that the fungus likes to live on to this huge volume of water where the fungus could easily be present, but you just might miss it by chance because they're little tiny bits of fungus floating around. And we have to try to get a handle on how much do we need to sample to detect it at low levels or medium levels or high levels.
While Grear's main involvement is in testing, in the event of an outbreak on American soil, testing efforts would become an integral part of our national response, and I asked him to dive a bit deeper into this process. I just want to ask you, when and if the pandemic does show up, what specific things would you do to work with the National Bsal Task Force to continue testing, to monitor the situation? How would that play out?
So there's a rough outline for a response plan, and it would involve a lot of calls, getting on the phone, video conferencing with the land managers, the state agencies or federal agencies where it was first found, to make a plan. And very likely it would involve mobilizing quite a few biologists to go out and do the testing, do our best to assess the status of the introduction. And we think it will be an introduction at this point if it comes. It's identifying where are the other places that we're most concerned about that are either nearby or somehow connected to where we find it. So, the analogy that perhaps you've heard with people talking about the initial stages of testing or trying to do control for the coronavirus pandemic is trying to deal with it like a forest fire, where you both have to deal with the main part of the fire where the introduction might occur, but also the trying to get a handle on where the sparks might go so they don't cause more fires. It would be that very similar concept of a response where there'd be a lot of attention paid to where it was first detected, but also an effort to try to prioritize, sample, and test areas where it hadn't been detected yet to get ahead of it.
I'm guessing, at least I'm assuming at that point, there would be like an increase in probably funding and resources being directed towards this effort, correct?
I would anticipate that, yes. It's been, within the USGS, within the fish, well, really within the science agencies, within Department of Interior and the management agencies, and as well as, right. So I anticipate that there would be attention funding to respond to a Bsal introduction or detection. It's high enough, I think, on conservation radars, on federal management agencies, on state management agencies' radar of priority things that there would be the resources put together to respond.
Before my conversation with Grear ended, he expressed that while he is happy with the combined efforts to search for and mitigate Bsal, he was adamant that it was important that national attention needs to be continuously sustained, especially in the absence of disease detection.
I guess I will maybe get on just a little bit of a soapbox about the difficulty of sustaining the kind of surveillance effort that we probably need to do, along with all the other research activities going on within the Bsal world and with the task force, especially when you don't detect something. It's quite easy to get attention to a disease emergence, a disease event, especially if it's something that's big and apparent and causes lots of sickness or nasty symptoms. But the challenge that we, as a disease, especially as a wildlife disease community, face is maintaining the effort at the same time as risk is only dropping a little bit. We're certainly taking actions to try to reduce risk, but the concern is always that the attention and the funding for doing testing necessary to get ahead of a pandemic will dry up before that pandemic emerges.
Can you tell me a little bit about the challenges you're facing then? What are some of the problems that you're seeing in having that funding and that level of testing sustained?
In our case right now, I think it's not as big of a detriment as much as it is. I look at what we did over the past 4 years and would love to continue at that effort. We've been, I think, fortunate that the various stakeholders in conservation, salamander conservation, especially in North America, have really made a lot of noise about the risk of Bsal. And so we continue to be able to do active work towards, and I say we, both at the USGS and with partners at different agencies and NGOs and universities. We have enough support funding to do active work towards trying to find preventive actions or responsive actions, even at this point anyway. So having seen a few other instances of wildlife disease where attention and money has, essentially completely dried up after the problem either went away or never emerged, we're, I think, in a relatively optimistic place for both working to prevent and respond to Bsal should it show up in North America or become really apparent in North America.
Correct me if I'm wrong, you were saying that you were concerned about the interest level dropping before we had a chance to get on top of it.
Yeah, and at this point, that concern hasn't dropped off so significantly that it's causing problems for, continuing to trying to find solutions. But that is always, especially in the world of wildlife disease and with where the impacts are to, non-game species, that's always a concern that the interest will drop off before we either have a chance to find the solutions or, before the problem really emerges because of it seems like it's not there.
I also asked Benji Jones how he felt about sustained testing efforts, and he too expressed satisfaction with the current testing efforts thus far and confidence in the continuation of them, and also a national response plan.
From the perspective of scientific strength to fight this off, I feel like we're in really good shape. I mean, the team that's working to combat Bsal is incredibly impressive. They seem really prepared. I think, like, yes, they have scars of working with BD, so they don't want to screw this up. But it really shows. I mean, like, I was just really impressed to see the kind of work that they're putting together and the fact that they're meeting somewhat regularly and they have like this many-page long emergency response plan. I mean, that's pretty rare to have. So they seem incredibly prepared. It seems like the biggest issue would be with the pet trade, just like what regulations look like there. But I don't think that's something that is easy to control. And I don't really understand what the full regulations that are in place are now. But I think like the greatest risk is that it would come to the US via the pet trade, and that's how it would spread, even within Europe as well. So if there are holes in terms of like holes that could lead to the spreading, that's what it would be.
With a solid foundation of first-hand experience with past pandemics and a soon-to-be-released document describing all the research that Grear and those that have worked with him have conducted, it appears we are well poised to respond to the fungus when it finally arrives.
I guess I just leave with, part of the, again, that I'm not totally pessimistic. I'm a little bit optimistic about our place, both because we're continuing to do work, and the Bsal task force has done a really good job of keeping the various pieces of research and management action in a centralized platform so that we can, even if one group or agency isn't able to do a whole lot, we're able to share the information we learn.
And I'll be talking about that information on the next episode in the series when I sit down with Dr. Jake Kirby, who is the former chair of the National Bsal Task Force. I would like to personally thank both Daniel Grear and Benji Jones for their contributions to this story and speaking with me on this episode in the podcast. I'd like to extend a hearty thanks to our podcast producer, Eric Hoffner, and also to Rhett Butler. Watch for a new edition of Mongabay Explores every 2 weeks, in between episodes of our flagship podcast, the Mongabay Newscast. Special projects like this are made possible by our Patreon supporters. Mongabay is a non-profit news provider, so we rely on the generosity of our listeners, readers, and friends. To add your support, head to patreon.com/Mongabay to learn more. Keep up with all of Mongabay's news from Nature's Frontline at mongabay.com, or get updates via Twitter, Facebook, and Instagram, where our handle is @Mongabay. Thank you once again, and we'll be back soon with another episode of Mongabay Explores.